But is it possible to administer the nanots, given the fact that you can scale up to 100 times more than what you've done and have no effect? So you can be hitting multiple things at the same time when it comes to cancer.
Well, actually, that's not just a smart question. It's almost like you were eavesdropping on a conversation we had with our team this morning. So there
Hmm.
is a debate internally in our group as to whether we're going to make individual nanots against these individual inhibitors and then combine them in one syringe to take out multiple inhibitors at the same time? Or does it make more sense to create one nanot that has capture agents against multiple targets on that nanot? And our team is divided. I
Hmm.
feel that, well, I'm going to just not say who takes what position, but I'll say
Yeah.
one position in our team is that you maintain combinatorial flexibility by only making one nanot against one target. And then you can combine them in the syringe at multiple, you pick the ones you want, put the concentration in the way you want. And as your disease, with different diseases or even different concentrations of target in a given patient, you have flexibility. The
Hmm.
other point of view is that from the FDA's point of view, uh, individual nanots are separate products, whereas a single nanot that can take out multiple targets at one time, that's one product. So we can potentially go through the FDA approval process faster with a nanot with multiple capture agents targeting multiple soluble targets than we can taking multiple nanots through the approval process.
Hmm.
Um, we need to get more guidance from the FDA. We're working on that to figure out what's the fast track here. I will clarify one point, which is nanots are not non-toxic because well, first of all, we haven't seen any toxicity dosing them at
Hmm.
100 times the planned dose. That doesn't mean there isn't any toxicity, but normally it would have shown up by now with a drug. That level of safety in preclinical studies is really pretty remarkable. We're not actually surprised because, first of all, the materials we're using are safe. Secondly, we're not taking out anything that belongs in the body, at least not at this concentration. So we're taking something out that's tumor generated that doesn't belong in the body at all. And the third reason we're not surprised by the safety profile so far is this concept of shielding. So nanots are adsorptive particles. They're basically like a sponge. They soak up something from blood. And to do that, they use a capture agent. Now, that capture agent would be bioactive if it could touch a cell surface. We only want it to deplete soluble targets in blood. We don't want it to do anything to cell surfaces because that's where the vast majority of drug side effects come from. So we have a concept which is in our patents, it's patented, and we just call it shielding. And the idea is the capture agents are nested down in the middle of this porous forest of shielding molecules. When the nanode as a whole touches a cell, the cell doesn't see anything except these inert shielding molecules. But the soluble targets diffuse through that shield and get captured by the capture agents on the core of the particle. So it's a really neat geometric, non-chemical, geometric solution to the challenge, this fundamental biological challenge. How do you take out a soluble target that's driving a disease and not disturb the membrane form of that same target that's essential for health?
So
Check out the full video now and subscribe. It's free.